Hello, we are Tyler and Jennifer, and we will be presenting variants in EXO-SE9 disrupt the RNA exosome and result in cerebellar atrophy with spinal motor neuronopathy. The exosome has 9 subunits responsible for the processing and degradation of mRNA. EXO-SE1 through 3 forms the cap of the exosomal ring for RNA recognition and binding. EXO-SE4 through 9 forms the core, a hexamer channel that the RNA passes through. Recessive variants in EXO-SE3, EXO-SE8, and RBM7, an RNA binding motif protein and exosome cofactor, can cause neurodegenerative disorders. This paper studies 4 individuals who carry autosomal recessive traits of EXO-SE9 and are affected by symptoms related to pontocerebellar hypoplasia type 1. Morpholino knockdown and CRISPR-Cas9 zebrafish models were created to explore the mechanisms of exosomal disease and showed similar defects in the cerebellum and motor neurons. To confirm the presence of EXO-SE9 variants in 4 individuals and their parents, exome sequencing and Sanger sequencing were used. The variants were isolated and analyzed against databases. MicroRNA analysis using an SNP probe was used to isolate the region of interest. The Lu14Pro mutation was discovered in all 4 individuals and seems to cause a disruption in the first alpha helix of EXO-SE9. This mutation seems to cause motor neuronopathy, muscle weakness, respiratory impairment, and cerebellar atrophy. Individual 2 also had an ARG161 mutation that stops the protein prematurely. This seemed to contribute to more severe symptoms, such as congenital fractures and respiratory failure and death at 15 months. Haplotype analysis was performed to identify shared regions of homozygous variants from the individuals with the specific homozygous EXO-SE9 variant. These were used to assess possible shared ancestry. To evaluate the phenotypic extent of exosomal disease in the 4 individuals, electromyography was used to assess motor neuronopathies. Brain MRIs taken at different stages of development showed progressive cerebellar atrophy. Muscle biopsy demonstrated denervation. Individuals had developmental issues, skeletal fractures, and muscle weakness. Genetic testing for other diseases that caused similar symptoms were negative, showing that the EXO-SE9 variants were solely responsible for these phenotypes. Cultured fibroblasts from affected individuals with variants in EXO-SE3, EXO-SE8, EXO-SE9, and RBM7 were immunoblotted alongside control fibroblasts. Figure A shows individuals with exosomal mutations all had reduced amounts of EXO-SE9, as shown by the lighter bands. Reduction is most pronounced in cells with EXO-SE3 and EXO-SE8 mutations. Other components of the exosome complex were also reduced. Actin was used as a loading control, which ensures that all lanes have the same amount of sample. Figure B shows blue native page that demonstrates a reduction in exosome assembly in the fibroblasts of affected individuals, as shown by the letter bands to the left, as compared to the thicker bands of the control. GAPDH acted as the loading control in this. Figure C shows an immunoblot of skeletal muscle extracts from individual 2 and highly reduced amounts of EXO-SE9. Mutations in exosomal subunits can impair exosome function and lead to disease. 3 possible mechanisms by which exosome function is interrupted by amino acid changes in exosomal proteins are disruption of complex assembly, as shown in Figure A, where the complex just cannot assemble. Disruption of interactions with RNA targets, as shown in Figure B, where the RNA is not able to bind to the complex. And disruption of interactions with exosome cofactors, as shown in Figure C. So, like the RBM7 protein, that would not be able to interact correctly with the exosome complex.
In order to look at the effect of the reduction of EXO-SC9 expression or variation on early development, zebrafish embryos were used as a model organism. Zebrafish have similar genetic structure to humans, with 84% of the genes known to be associated with human disease having a zebrafish counterpart. As vertebrates, they share the same major organs and tissues as humans, and their genome has been fully sequenced to a very high quality. In order to reduce the expression of EXO-SC9 gene, embryos were injected with morpholino oligonucleotide, a type of oligomer molecule that blocks access of other molecules to small specific sequences of the base pairing surface of RNA, knocking down their expression. In this experiment, the morpholino blocks access between exon 1 and 2 of the EXO-SC9 gene region, leading to aberrant splicing and retention of an intron, reducing its expression. Due to a recent criticism of the specificity of morpholino and suggestions of off-target effects, CRISPR-Cas9-mediated mutagenesis was used to induce target-specific variants in the EXO-SC9 transcript. CRISPR-Scan was used to identify a target site in exon 3 of EXO-SC9 in zebrafish. The sgRNA templates were generated and consist of an oligo of the T7 promoter, 20 base pairs of target sequence, and 20 base pair sequence that overlapped to a generic sgRNA template. The sgRNA and Cas9 protein were injected into the embryos and induced variant mutagenesis of EXO-SC9. The embryos that underwent morphine knockdown were labeled as morphins, and the embryos that underwent CRISPR mutagenesis were labeled as crispins, and were organized by their severity of knockdown or mutation, with higher severity indicating a higher amount of knockdown/mutagenesis. This severity was determined by RNA isolation by embryo lysing or PCR cloning, followed by reverse transcription PCR and Sanger sequencing. Looking at a morphological scoring picture of zebrafish morphins and crispins of varying severity, it is clear that the more severe the knockdown or mutagenesis, the more intense the developmental defects the embryo will have. Compared to a normal control embryo, mildly affected embryos had smaller heads and eyes, whereas severely affected embryos had extremely small, sometimes absent eyes, very small heads, and truncated bodies, all signs of developmental defects in the brain and muscles. The phenotypes and distribution of severity was consistent between both the morphine and crispin embryos. Whole mount immunofluorescence, or the fixing of a fluorescent antibody to its target for imaging, performed on morphins and crispins with an antibody against the neuronal marker HUC, shown in green, showed that the brain fails to properly develop. Compared to the control, the morphins and the crispins commonly had a misshapen midbrain, an absent cerebellum and hindbrain, and a lack of a posterior cranial nerve. Using antibody against synaptic vesicles 2, shown in green, and neuromuscular junctions A bungarotoxin, conjugated to Alexa Fleur 594, allowing it to fluoresce red, showed that these junctions developed abnormally in the morphins and crispins, with the junctions appearing closer together and the motor axons failing to migrate properly to them, indicating a primary pathfinding defect of motor axons also seen in EXO SC3, 8, RNB7, 19, and SMA variants. It is important to note that they could not find any antibody that successfully identified any of the exosome components in zebrafish to prove that there was indeed a reduction at the protein level. In conclusion, the clinical evaluation of the 4 individuals shows that the variations in the EXO-SC9 gene cause cerebral atrophy and motor neuronopathy consistent with PCH1-related disorders, which are also seen in EXO-SC3, 8, and RBM7 variants. Using zebrafish as a developmental model, it was seen that the expression, knockdown, or induced variation of EXO-SC9 gene leads to brain and muscle developmental defects consistent with the affected individuals and other gene variants. This consistency displays the effect the EXO-SC9 variants has on development, but also shows the usefulness of zebrafish as a model organism. In the pimal recreation of the exosome complex shown on the right, the light blue at the bottom represents the catalytic subunit of DIS3. The gray in the middle of the structure represents the 6 barrel proteins EXO-SC4 through 9. The cap subunit of EXO-SC1 through 3 is shown in orange, green, and yellow at the top. The blue subunit at the top is EXO-SC10, and the white residue is the RNA, which can be seen interacting with both the 3 cap proteins as well as DIS3. It isn't known how variation in these genes affect the exosome to cause these phenotypes, but looking at its structure could point to some clues. Variation in the cap subunit EXO-SC3 could lead to inability of RNA to enter the barrel. Variation in the barrel subunits EXO-SC8 through 9 could lead to inability to bind the RNA. Variation in the cofactor protein, RBM7, could lead to RNA mistargeting, and all variants could lead to instability of the exosome structure. Now on to our outstanding questions. First, further studies are required to determine the effect of variant mutations in exosome subunits on overall exosome structure or RNA targeting. Second, because they were unable to identify an antibody for exosome components, did morpholino gene knockdown or CRISPR-Cas9 mutagenesis of EXO-SC9 result in a reduction of the amount of exosome proteins in the embryos? Third, how strongly are the phenotypes affected by morpholino gene knockdown or CRISPR-Cas9 mutagenesis off-target interactions? And finally, further studies on neuronal cells of infected individuals or zebrafish might reveal neuron-specific alterations which could be targeted in future interventions. And here are our references.